By Kirsten Thornhill. Trichomes are primarily created on the flower, leaves, and bract of the cannabis plant. They have crystal-like, surgery appearances. A trichome is almost like an accessory on the surface of the cannabis flower, producing and holding the plant's terpenes and cannabinoids. Trichomes are the non-glandular and glandular hairs found on the surface of plants. They are responsible for producing cannabinoids and terpenes in the cannabis plant. The word trichome itself derives from the Greek word trichoma, meaning growth of hair. As these hairs are micro in size, you may need to zoom way in to see the resin blanket that coats cannabis buds to be able to see the fields and hairs of trichomes. Although it's likely you weren't familiar with trichomes, you've probably seen trichomes before. These tiny hairs cover your cannabis, perhaps giving it a surgery shine, maybe even a sticky feel. What are amber trichomes made of? Trichomes can consist of scales, hairs, glandular hairs, and papillae. They grow on the plant appendages, such as cannabis and algae. To be more cannabis-specific, trichomes contain resin glands that are responsible for making terpenes, CBDA, THCA, and other cannabinoids well known to cannabis. What are the functions of amber trichomes? In the cannabis plant, trichomes can function as a defensive mechanism. When female cannabis plants start to produce flowers in the wild, they often become vulnerable and susceptible to various animals and insects. Trichomes can serve as a deterrent for animals due to their bitter taste and intense aromas, making them unappealing to our friendly creatures. Trichomes can also function as a protector, keeping their plants safe from strong winds and varieties of fungal growth. During cultivation specifically, trichomes can act as a deterrent to keep away pests, predators, and/or environmental stressors on the plant. They can assist in keeping the plant's surface level humidity optimal. This will help keep the plant from drying out and keep it in production mode for producing terpenes that repel insects and pests. The small bulbous glands can also act as a trap, confining small insects. Trichomes really do play a key role in cannabis, as they can be huge helpers when growing in difficult climate conditions. A layer of resin, thick or thin, can offer wonderful weed protection against damaging winds, from deserts to blizzards. Trichomes can even be thought of as sunblock for the cannabis plant, as a protectant from the sun. They can protect the plant from harmful UV rays. What are the different types of amber trichomes on cannabis? Trichomes can exist in various shapes and sizes. However, there are three types that appear most often on the cannabis plant. 1. Capitate stalk trichomes. This type of trichome ranges from 50 to 100 micrometers wide. They are much larger and can be seen by our naked eye. Structure-wise, capitate stalk trichomes are composed of hypodermic and epidermal cells that work to build up a basal cell, attaching to a larger glandular head. This glandular head is held together by a waxy cuticle layer, serving as an epicenter for cannabinoid and terpenoid synthesis. 2. Capitate sessile trichomes. Capitate sessile trichomes are slightly bigger and contain both a stalk and a head. These trichomes are more abundant than their bulbous sister, but can't quite keep up with the size of the capitate stalk trichomes. 3. Bulbous trichomes. These trichomes are the smallest of the trichome families. They appear on the surface of the entire plant. How are cannabinoids created in trichomes? Cannabinoids are created within the trichome cells through a process called biosynthesis. During this process, enzymes catalyze a series of chemical reactions, producing complex molecules from smaller molecules. 3 important steps for this cannabinoid biosynthesis include: molecules bind to one or two molecule substrates, then bind the substrates to one another. They then pass the connected substrate further down the assembly line to another set of enzymes that further process it. This cycles the small molecule through further changes. The most well-known enzymes responsible for producing cannabinoids are cannabichromenic acid, CBCA, synthase, cannabidiolic acid, CBDA, and tetrahydrocannabinolic acid, THCA, synthase. These enzymes take the central cannabinoid precursors, cannabigerolic acid, CBGA, and cannabidivarin acid, BGA, and convert them into acidic cannabinoids, including CBDA and THCA. Next up, these cannabinoids may be altered through further decarboxylation. When this happens, it loses a carbon atom and 2 oxygen atoms. It then releases carbon dioxide. THCA and CBDA decarboxylate into THC and CBD, respectively. This process must happen to exhibit their characteristic effects. This can be considered a degradation process, as it does not require enzymes and occurs only after the plant has been harvested. How are amber trichomes produced? Within the trichome, the cannabis synthesis begins as the cannabis plant itself moves into the blooming phase. As the plant begins to bloom flowers, trichomes also begin to form along the outer surface of the plant vegetation, transporting vacuoles and plastids from the stalk into the glandular head. Cells within this glandular head soon metabolize, forming precursors for what will soon become cannabinoids. The concentration and rate at which cannabis produces individual trichomes depends on both environmental and genetic factors. Plants containing higher concentrations of trichomes may not always produce the highest concentrations of cannabinoids or terpenes, so to speak. For instance, variables such as UV light will greatly affect cannabinoid and terpene profiles within the trichome head. In a general sense, plants that receive more light may produce higher concentrations of cannabinoids, although, in some instances, they may react depending on their specific strain. How is the life cycle in amber trichomes? A trichome's life cycle is very similar to that of the mother cannabis plant, making it a super value for farmers to monitor. The life of trichomes can be compared to parabola, where the apex is the point at which maturation goes beyond and degradation starts. Amber trichomes typically display maturation on this parabola through opacity changes, from a clear translucent state to a murky white. Thanks for watching. Please subscribe.